Good afternoon and welcome from the land of the Darg and the Gundagar. Another beautiful day here in the Blue Mountains. Welcome to the first Science at the Local for 2021. Sadly, we are online, but we are here. So first, we'd like to pay respects to Aboriginal people and elders, past, present, and future. And again, acknowledge the Blue Mountains as the land of the Darg and the Gundagar people. My name's Kevin Joseph, and I'm here with Hamish Clark, and we're the organizers of Science at the Local. I won't take a lot of time because we want to get to Sheree, our speaker, but just a reminder, as we go through the talk, there is a Q&A button down at the bottom of your screen. If you have questions at any point in the talk, please just type your question there. You don't have to wait till the end. And then Sheree will get to the questions at the end of her talk. I'll throw it over to Hamish to introduce our speaker. Here you go, Hamish.
Thanks very much, Kevin, and nice to see all of you remotely. It's great to be here. I wish we were here in person, but the benefit of the technology is we get to bring in star-studded speakers from all around the world, including Sheree today. Before I introduce Sheree, I will also thank our fundings partners or supporters, Inspiring Australia, who've made possible a lot of what we do. So thanks to them, and check them out. They support a huge range of science communication activities in New South Wales. So Dr. Sheree Yau is a microbiologist who researches the life of marine algae and their viruses at the Banyuls Oceanologic Observatory in France. After doing her undergrad studies at the University of Sydney, she did her PhD at the University of New South Wales on the microbial communities of Antarctic lakes, which contain seawater that's been separated from the ocean for thousands of years. She then moved to France and then from there on to Spain to continue her work on microbial ecology, studying how microbes live in their natural habitat. In Spain, Sheree worked on new methods to understand viral dark matter. And since returning to France, she's continued to work on one of the biggest questions in microbial ecology. How do we link the astronomical number of viruses in the environment to their host species? So please, a big digital round of applause and welcome to Sheree.
Thanks for the very kind introduction, and thanks for the invitation. I feel very honored. And yes, I benefit from lockdown that I can come to Australia virtually, even though I can't come in for real life. Okay, so I'll just go straight to it.
Great, we can see all that, thank you.
Great, I'm going to put on a laser pointer. Okay, so I'm talking to you today about my work, marine algae and their giant viruses, and why they matter. So I'm just going to flash through this. This is my bio in a nutshell. Australia to France to Spain, back to France. So here I am right in the south of France, working on algae and viruses for the French government. I work for the CNRS. Okay, so an overview of what I'll be talking about. Why do marine algae virus interactions matter? I'll talk about Ostracoccus and its viruses. So this is my favorite model system. And I'll tell you, what do we know about Ostracoccus and virus interactions, including some fresh off the press new data. Okay, so why do algae virus interactions matter? Well, all cells, so all living things are infected by viruses, not just us. They're ubiquitous molecular parasites. And so all cells have come up with ways to fight viruses, and in turn, viruses find ways to fight off these defense mechanisms. So this constant interaction in the phenotypic level, so this arms race leads to reciprocal selection on between cells and their viruses and coevolution of the two. This is happening at an individual cell level and with their individual genes, but then also translated to population level and under the constraints of the environment. So we're all living this as we speak. So how do single cells defend themselves? I mean, we're multicellular organisms. The best models that we have are for bacteria. So basically a cell can decide to stop the virus at the door. So to stop virus recognition by changing its lifestyle, or there are a whole bunch of molecular mechanisms. So restriction modification is RM, these very famous CRISPR-Cas systems, and many more being discovered that stop viral replication inside the cell. And if all else fails, the bacterium can decide to kill itself and stop viral propagation that way. So that's called abortive infection. But on the other hand, for things like, so I'm talking about algae, but this is even larger, so protists or eukaryotic single-celled organisms, so our relatives, we don't really know how they defend against viruses. So there are some examples known of them switching lifestyle, so going into dormant forms or becoming flagellated or grouping into colonies, and that helps protect against viruses. There's a sort of new system being understood of these viruses that stop viral infection, but it's really just starting to be discovered. And there's some evidence that cell death, a cell might kill itself if it's infected. It's not totally clear whether this is to help the cell or to help the virus. So basically, there's a lot of unknown about how unicellular eukaryotes or protists defend themselves against viruses. So why is this important? So in a very, very big scale, in the oceans, CO2 is fixed into organic matter by phytoplankton. And in the classical food chain, the zooplankton will feed on phytoplankton, and all the way up the grazing chain, so they'll be fed on by little shrimp and fish and up to big charismatic organisms. So it's been a bit over 30 years since the discovery that viruses are the most abundant biological entities in the planet. So for every cell that's in the ocean, there are about 10 viruses. And by killing cells, viruses direct all this biomass, all the phytoplankton are doing, away from or down the food chain, away from high organisms, into just dissolved fraction as just free organic matter. And so this can also act as a carbon sink. So carbon can sink down to the deep ocean and be stored for thousands of years. So just to put this in a big perspective, CO2 in the atmosphere, there's about as much organic carbon just floating around in the ocean as there is CO2 in the atmosphere. Phytoplankton perform about 50% of planetary carbon fixation or photosynthesis, so as much as what all the plants are doing on the land. But of course, we don't see these massive forests in the oceans because the turnover is so high. They're being fed upon or lysed fairly quickly. But the bounds on this rate of viral mortality of phytoplankton is not very well understood. So to get at that, we need to know more about the biology of how it works. How do they infect their hosts? And so I introduced Ostracoccus and its viruses as a model system. So Ostracoccus is famous as being the smallest eukaryote. So here's a picture of the cell. So a eukaryote, as I said, is a cell like us. It has a nucleus. They're different from us because they have chloroplasts. And you notice that it's very tiny. It's only about a micrometer across, which is the size of small bacteria. And here you see the cells being infected by viruses. So these are called praseoviruses. They're in the giant virus family. They're relatively small for a giant virus, but for their tiny, tiny cells, it's relatively huge to have a virus like a tenth of your size being stuck on the outside of it. These algae are widespread in the environment. So basically just all these blue bubbles around the world are places that you can find algae like Ostracoccus. And this pie chart is just to say that praseoviruses are also widespread and abundant So when you look at total virus communities in marine waters, they're a fair chunk, perhaps the most abundant. All right, so just an aside, well, how much do we know about viruses of algae in the world? Well, this tree is a tree of all marine unicellular algae. So every colored branch and all these little cartoons are different big groups of marine algae. So you see they're very diverse in the sense that, okay, here's animals over there. So our branch is the only one that does not have photosynthetic organisms in it. So the little stars represent very abundant groups in the marine environment. And the little hexagons are the viruses that are known to infect them. So basically, you see a lot of these red hexagons, therefore the NCLDV. So these are giant viruses, just like the parasinovirus I work on. And there are some RNA viruses known, a few single-stranded DNA viruses known, but we know most about these giant viruses. And that's not really saying a lot that we know most about them, because, well, how many viruses do we know about? To try and put this in perspective, this is a bit of an old slide. I did this in 2018. I just went to the website of the ICTV, which is the International Committee on the Taxonomy of Viruses. They control what's recognized as a species. And so back then in 2018, there was about 5,000 species of viruses known. Well, is that a lot? As an aside, this was a cool study done to try and just know how many viruses infect mammals. So everyone's heard of bats and bats carrying viruses. So what they did, they just got a bat. They did PCR. Now we all know what PCRs are for all the virus families known. And this bat had 58 different viruses. So if you say, well, one mammal has 58 viruses. Well, if we took all the mammals in the world, all the mammalian species, it's about 5,000 of them. If all the mammals had 50 viruses, well, we come up to over 300,000 just mammalian viruses. So knowing 60 algal viruses that span the whole tree of life is not really that much, but that's what we've got to work on. Okay, so back to my story. This is what Ostracoccus species look like. So I work a lot with genomics, and the reason why is because so I introduced Ostracoccus as the genus name. It has 4 species known, so Ostracoccus tori, Ostracoccus lucimerinus, an unnamed species, and Mediterraneus. And these are what they look like under the electron microscope, and they look the same. They're just green blobs. You cannot distinguish them by microscopy, but when you look at their genomes, they're exceedingly diverse. So what's exceedingly diverse? If we compare Tori and Mediterraneus. So these are the 2 species I'm going to talk about today. So remember them. They are 72% identical in terms of their protein sequences and average amino acid identity. So you heard that humans and chimps are 99% identical. Well, this is humans and chickens are 23% divergent. So about 70% identical. So between these 2 green blobs that look very similar, they have as much genetic distance as humans and chickens. So it's a lot. Genomics also revealed these 2 funny chromosomes in Ostracoccus tori. So it's got 20 chromosomes. These are all laid out here. You can see immediately that chromosome 2 has this big bunch of blue, and chromosome 19. So I just ruined the story. We think that chromosome 2 is the sexual chromosome, and 19, we didn't know what it was. So we called it the small outlier chromosome, or the SOC, for want of a better name. And so just remember Ostracoccus tori, Mediterraneus, and the small outlier chromosome, the SNOC. So what do we know about Ostracoccus and virus interactions? One thing that happens reliably when you grow Tori in culture, so this is a curve of it growing, it looks green, nice and healthy. When you infect it with viruses, the population crashes. But very quickly, you get resistant cells growing up within 3 days. This sort of pattern of becoming resistant to viruses is also seen in completely different algae. So you don't have to remember all their names, but that you see the same pattern of the normal growth or death by virus and growing back. So the idea is to look at Ostracoccus as a model for understanding this sort of pattern. The other thing that we observed was that resistance to virus also leads often to coexistence. So what does that mean? Here you see the cells growing, and at the same time, viruses are growing. So we call this a resistant producer, for also want of a better term. And that seemed odd. So the cells, the culture wasn't dying, it was still growing, but the viruses are still growing too. And this was really surprising. We saw this in Ostracoccus mediterraneans. It was isolated from the environment in this state. So you see the green line is the algae growing, and the purple triangles, the virus is growing. And that it's stably like that since 2009 in culture. So how does that work? How is the virus without killing the culture? So these are just more cell pictures with some viruses inside. So we take a resistant culture and just look with electron microscopy very hard. Most of the cells were looking nice and healthy like this, but a certain proportion, you could see little viruses growing inside them. And it was about 1% of cells in the Ostracoccus tori resistant culture that were visibly infected. So the same thing was observed for Ostracoccus mediterraneus. Had about 1% of clearly infected cells. So this was our response, that a minority of S cells or susceptible cells are able to sustain the virus production. So most of the cells are nice and healthy, just a minority of the cells in the culture are dying off and growing the virus. Okay, so how does that work? To answer that question, we have this complicated schema of experimental evolution. You don't have to follow all of these details. It's just to show that we start with one clonal algae. We keep some as S, so susceptible. They don't see any virus. And then we have many parallel lines. So in this case, 38, that were exposed to virus, they died, and they became resistant. And then what we do is try and compare the genomes and the transcriptomes. So this is all of the genes that are being expressed in terms of becoming RNA between resistant and susceptible states, between R and S lines. Okay, so the first thing, we look at the genome. Susceptible Ostracoccus tori, when it becomes resistant, we could see this change in pattern. So this lovely image, all the chromosomes of different susceptible lines separated on the gel. And you see in our susceptible S lines, there was no difference. But in the resistant lines, there was a notable change, mainly here at the chromosome 19. So that little chromosome that we called the small outlier chromosome changed in size. The same thing was seen the other way around in a different species in Mediterraneus. When resistant cells became susceptible, we see a deletion in this, the equivalent of the small outlier chromosome. Now we could see which bit, it was the end bit of the chromosome came off. And there were a few genes that were unique in this region that were lost. Okay, so we have susceptible to resistant, resistant to susceptible cells lose or gain bits of this funny chromosome. The other perspective we took was to take strains from the wild and compare them to their reference. So basically here, anytime it's white, it doesn't match to our reference genome. And you see it only happens in this chromosome 19 in the sock, that there's no matches. And this is because every individual strain had an individual unique chromosome 19 or sock. There's just a few bits that were common. So it is a very variable, hypervariable chromosome. What's more, looking at these strains, we could test them against a panel of different viruses. So these are all different persinoviruses, and ones means they are infected and 0 means they're not infected. And you see that there's a spectrum of resistance to different strains of viruses, and this is directly correlated with the length of the chromosome 19 to the sock. So out in the wild, this chromosome is related to resistance and susceptibility. So not only does the size matter, it also seems what you're doing with it matters. So if you compare the pattern of expression of this chromosome in Ostrococcus tori, resistance lines express this part and it's not in the susceptible, and vice versa. So you had this on and off sort of switch on half of the chromosome. What are those genes doing in this resistant part? Well, they're mainly to do with sugar transport, methylases, and a lot of things that are mobile elements or transposons. So it's a big mess. There's a lot of things going on. And just to show that the same sort of pattern of half being off and half being on was seen in Ostrococcus mediterraneus. And they also have very similar complements or types of genes. You notice that the unknown is the biggest category. So we've got a lot of work. And so, as I said way in the beginning, we know that bacteria, as their first line of defense, is to stop viruses getting in. So the very obvious question was, can the resistant cells attach viruses? And in the case of Ostrococcus tori, this shows that resistant lines don't attach the virus anymore. So that's probably how they've become resistant. They've changed something on their surface, so they're no longer seen by the virus. And I thought, oh, Mediterraneous is the same in all other ways. So I, anyway, just tried the same experiment recently. And well, it seems that it's not the case. The amount of viruses that can attach to resistant or susceptible Ostrococcus mediterraneus is about the same. So it's doing it in a different way. And as a sort of cherry on the top, how do we try and link all of this back to maybe what's going on in the environment? So this complicated image is just saying that, all right, so if susceptible and resistance can switch back and forth spontaneously, we don't know if it's spontaneous, but just imagine it's spontaneous. When there's no virus, you kind of don't see anything. If you do have a virus, you kill off most of the susceptible population. But if some resistant cells are always switching back to being susceptible, this is how you can get to a stage of stable coexistence where just, you know, that 1% or a certain proportion of resistant cells are always just losing their resistance and allowing the virus to continue to be propagated in a population. So both of them can grow together. So the take-home messages, so we need to find out what's going on in terms of the molecular mechanisms or the genes involved in determining host virus specificity in eukaryotes in general or protists, and particularly in algae-virus systems. Because if you don't know how it's working on this molecular level, it's hard to predict what's going to happen on the population and then the community level, and then all the way up until, you know, global carbon cycles. So our modest contribution here is that in this model organism, resistance or susceptibility is linked to a particular chromosome. Because we don't know how it's becoming hypervariable, changing in size. We don't know how it's switching between resistance and susceptible states. It seems to be spontaneous. I thought that it was just changing the surface of the cell It seems to be the case sometimes, but it's just one line of defense. There's probably others internal. And so it's the mechanisms are happening in terms of the genome, the expression. I didn't talk about metabolomic changes, but we've also got a little bit of data on that. We've got to work out how it works. And the net effect of getting resistance quickly is that, strangely enough, it can lead to coexistence. And the open question is, how common is this out in the environment? Okay, so with that, thank you for your attention. I hope that it wasn't too much of an info dump. All of this work is brought to you by Team Genomics of Phytoplankton, so the Genofi team. So we got our phytoplankton and the virus and our DNA. And this is where I work in aerial view. I'm in this lovely building here in front of the Mediterranean Sea. And these are my sponsors.
Thank you very much, Sheree.
Thank you.
If
I think
if
I'm
the
on
audience,
time.
perfect, extra points. If the audience is anything like me, their heads are spinning like a top. I have a few questions which I'm just going to pepper you with until the audience is brave enough to chime in too. Just a reminder to people, please use the Q&A button down the bottom of your screen to pop any questions in. The first one is, do you or your colleagues do fieldwork? Are you situated in that location for a reason?
Yeah, we're involved a little bit in field work. So there's this big consortium, Tara, with a sailboat that goes around the whole world. I've worked a little bit in Tara. We have done sort of field campaigns. So we isolated these different strains. We're still doing that, trying to isolate different viruses and algae from the environment. But I'm not doing a regular observatory. So there are actually in Sydney, you have this as well, these people who are actually going every month and collecting samples. So I'm not directly involved in that. So yes and no. I'm doing a lot of lab work.
Cool, thank you. Yeah. We've got a question from Jessica who's asking if you can tell us a bit more about your career journey and how you got interested in aquatic viruses in the first place.
Oh, okay. Yeah, so it's just serendipity. I thought, oh, metagenomics is cool. So that was just when I finished my honors and I was looking for PhDs, and I thought, sorry, oh, wow.
Can you explain what metagenomics is for any of our audience who don't know, please?
Yeah, so metagenomics is basically where you just grab a bunch of whatever directly from the environment. It can be poop, it can be sludge, it can be water. Seawater is nicer to grab. And you just take all of whatever's in it and sequence the DNA. And the idea is then to understand what all the microbes in the community are doing. So I saw that there was a PhD program at New South with Rika Cavicchioli on Antarctic lakes. I thought, wow, Antarctica, that's cool. And just it happened that in the samples that I was looking at, there were a lot of viruses, and then I got the bug of virus ecology. There's just so much diversity of viruses, and there's so much to do in it that, yeah, it's just a wide open field, right?
Before we go to the next question, Sheree, could you just tell us about your personal journey in terms of where you went to high school and how you ended up in France? And that might be also interesting for people to know.
Oh, okay. Maybe I won't go back to childhood. So I was born in Malaysia. Basically, I did all my schooling in Sydney from high school, and I went to I did a science degree at Sydney University. I looked around for PhD projects. I just saw one at New South. I maybe should have thought more about it, but there was, you're just sort of drifting along the path of least resistance. When you see open doors, something that's interesting, I went there. And after I finished while I was finishing my PhD, I saw a job ad in Banyuls in France, and I applied and I got it. And then I just, the inertia kept going. I wanted to stay there. And I just got the position with the CNRS last year. CNRS is like, I guess, the equivalent of CSIRO, but it's much bigger. So I'm not actually, we're attached to Sorbonne University, but I personally don't need to do teaching. I just do research. And this is I was very, very lucky to get this job. But I have to say, to get a job as a French civil servant, you have to go through a national competition. So every year they have a call, and actually anyone in the world can apply to join the French civil service as a researcher if you have a PhD. So if anyone wants to join the CNRS, in January you can write a project proposal and you could do it.
Get writing, people. Thanks, Sheree. We've got a question from Deborah, who asks, are the Ostrococcus species important commercially?
Not totally, no, because I guess I'm trying to boost that aspect. They're really little. So people like using algae that make a lot of oil or do something sexy like that. They do. I'm a bit more detached from the biotech side, though I'd like to get more into it. But as far as I know, they're not heavily exploited. But I would like them to be more exploited for these ends.
Thank you. Michelle asks, are there many viruses that exist in the deep ocean?
Yes, there are. So viral ecologists like to do just counting viruses. So looking at how many per milliliter. So let's say the surface, there's something like 10 million virus-like particles per milliliter. In the deep ocean, I think it's maybe like 1 million or just 10 times less. It's less dense, but there's still a lot of them. Yeah.
Thank you. Craig has asked, can you talk some more about the abundance of viruses? And also says, how strong is the specificity of viruses versus species? He thought the number of viruses calculated was very surprising.
Yeah, well, I mean, it comes down to the counting problem or the species definition. I mean, right now we're seeing all these different variants. Even what you call a strain is difficult because some people would like to say it's a strain only if it has a different characteristic. So, okay, we're seeing from a genetic point of view, Delta variant. Okay, we can see that it has a genetic change, but does it translate to actual change in its lifestyle? And at how many changes do you need before you're a new species? This is something that is very, very hazy with viruses because it's difficult to measure these sort of changes. But in terms of just the genetic diversity, it really is astronomical. It's just massive. If you think about how many just species of bacteria there are, all the bacteria have at least one virus. There's at least as many viral species, and they tend to be quite specific. So I'm not scared that my viruses in the lab will infect me. So they're fairly specific. Some have broader host ranges. Yeah, I can't even give a number. It's just astronomical.
Is the astronomical too conservative a field? Do we need to say that it's like microbiological, the size?
Yeah, maybe. Yeah, the astronomy. I mean, one of the things that people like quoting in viral ecology says like 10 to the 30 virus particles in the ocean or something like that. It's as many as stars in the sky, if not more. Yeah, it's a lot.
Wow.
We're really just like tapping at the very, very tip of what's out there. And so we like to steal the physics metaphor. So like saying it's viral dark matter. We've got physics envy, obviously.
Well, that's a perfect segue, actually, from the line in your bio and what you just mentioned. Can you tell us what viral dark matter is?
it's just a catchy term to say we can count little virus particles out there in the ocean, and we can say, oh, there's 10 million per milliliter, and what's the volume of water out there? And then we've taken it from the other perspective using, as they said, metagenomics. Let's just sequence all of the genetic diversity. Ooh, it's a lot of diversity, but what are they doing? So I can see a virus, I can sequence it. What is its host? We've got millions now of viruses known only from their genetic sequence, and we don't know what their host species are. So that's why we're calling it dark matter. It's sort of this just big blob of barely understood biology. It's not as unknown as dark matter, though. We know that it's biological.
Hey, can you tell us what dark matter is?
No, I don't know what dark matter is. It's maybe a false analogy.
Yeah, no, that's good. We like analogies here. Just a reminder, folks, if anyone has any questions, pop them in. We'll wind up shortly. I'm going to ask another one just because I can. Can you talk a little bit about the tools you use and how much that is changing, or how influential it is in the work that you do?
Yeah, so as I said, or I flashed in my bio, I do everything omics, so genomics, transcriptomics, and I've done a bit of proteomics. All of these technologies are being developed. So sequencing DNA, there's a few different ways to do it. There's long reads, short reads, higher and lesser throughput. So this really changes. For example, from my PhD, we were doing it with old school Sanger sequencing. We had very little information relative to what's out there now. And it's sort of still not enough. And we still have so much data that we don't even know what is happening with it. So it's really constantly evolving in terms of technology and what we can do. But at the same time, I'm using also very old school culture methods.
Cool, thanks. Well, Kevin, is there anything else you wanted to ask before we wrap up?
No, I think I was actually just looking at that image and I was just wondering, is most of your research conducted in French at CNRS, or?
So luckily, I get to write everything in all my like scientific articles in English, and I cringe when I have to write reports and things like that in French. Yeah, you do to get by in administration, do need to learn French, I think.
Okay, thanks. That just reminded me of all my not paying attention in French class as a student. Sorry.
Well, it's you can get by. It's just that you get by better if you can talk to the administration and write reports and read things. But it's not that hard.
You have another question. Oh, sorry.
There we go. Yeah, you go, Kevin.
Richard says, from a non-scientist, in the COVID context, viruses have a bad reputation. Do viruses have any positive impacts?
Yeah. Oh, that's a nice question. There's actually a paper written by Marilyn Rusknik called something like "Good Viruses." Yeah, we're kind of promoting the fact that viruses are really part of the circle of life. You imagine if we just removed all the viruses from this oceanic food web, maybe you would actually have less carbon export, maybe you would have less productivity in some ways because they're helping with the turnover. So they can have these net effects that are beneficial. And then on the completely evolutionary scale, we have tons of virus fragments in us. And so there's been a few cases where we have taken genes from viruses and they are part of our biology. So the really famous example is a gene to do with the mammalian placenta. I don't remember which gene it is, but apparently that was hijacked from viruses by us and we use it for our placenta development. There's other examples. There's a cool example of this grass that needs to have a fungus that is infected by a virus to live in hot springs. So they're also, in terms of generating genetic diversity, like a pool of genetic resources that we can exchange with as well. So their net effect can be positive.
Thank you. We've had another one come in, and we've finally come back to COVID. Are there any links? This is Thais or Thai, sorry if I'm mispronouncing your name. Are there any links between these viruses and others like COVID that are affecting humans now? Do those ocean viruses have a detrimental effect on the ocean organisms they interact with?
Yeah, I've looked at, so just off the bat, SARS-CoV-2 is a RNA virus. So it's not at all the most abundant viruses of algae. We actually, really weirdly, plants have no DNA viruses, or not the same sort of DNA viruses as I'm studying in the algae. And mammals seem to have a sort of equal mix of DNA and RNA viruses. So it's really, I've never seen anything like coronaviruses affecting microorganisms. They're just so far apart genetically. They're not even in the same ballpark. But the big themes of how they evolve and how we co-evolve, I think, are pertinent. Like we want to know how quickly we can sort of get to an equilibrium with the virus.
Great, thank you. Well, we might wrap it up there then. Once again, thank you so much for your time and your talk, Sheree. We are recording the talk, so you'll be able to catch up on your favorite slides later or share with friends who want to see on our YouTube channel. And we're hoping to have another event. Here we go, Kevin beautifully has shared it. End of October. We've got Mike Mainfield from UNSW talking about bioremediation. And we'll give you some more information about any other webinars we're planning on our Facebook page and our email list. So we would love to see you then. Thanks again, Sheree. Nice to see you all, and take care, everyone.
Thank you.
Thank you for
Sheree.
the invitation. Bye-bye. Lovely to be here.